In this video, you will learn about stereology, what stereology is, when it should be used, why is it not used as often as it should, and what happens then. Hi, I'm Alexandra Żuraw and I'm here to help you do better digital pathology. If this is what you're after, be sure to subscribe, click the bell below, and be notified every time I release new videos. So let me be honest with you, I have never done stereology. But Alexandra, how are you gonna tell us about stereology if you've never done stereology? Well, I invited an expert for you. This person has been doing stereology for years, he published on it and simply knows a lot about it. So after watching this video, you will know if you need it or not. So without further ado, let's meet Mike Staup and learn about stereology. Welcome to the channel, everybody. Today, my guest is Mike Staup, a PhD scientist who is actually my colleague. We work together every day, even though we work remotely. We chat every other day at least, and we both do image analysis. And even though I have been working in the tissue image analysis space since the beginning of my career, I have never even touched stereology. To teach myself what stereology is, I don't know, I watched a couple of YouTube videos, whereas Mike, he did everything I did, so handcrafted features, classical image analysis, AI-based image analysis, but he also did a lot of stereology. So with that short intro, I let you introduce yourself, Mike. Tell us about you and about your stereology background, because that's the topic of our video today.
So not too long ago, I was in the same place that you are, where I had never done it. It was coming out of grad school, really. So I did some digital image analysis as a grad student, and it was really unnecessary because the effect I was looking at was so profound. I could have just done a 5-point scale or even just shown images. But, as an eager young grad student, I wanted to do everything correct. So I did a objective, quantitative analysis of these CFOS, labeled cells in the various brain regions, and looking at the development of supraspinal motor areas. I actually ended up trying to do everything the right way, doing a lot of bad things that no one corrected me on because the effect was so profound. And so I developed a lot of bad habits. And then I went from there to my postdoc.
You mean stereology bad habits?
Just image analysis bad habits. Mm-hmm. So just quantitative bad habits. Mm-hmm. Because really, stereology is, I didn't do any stereology as a grad student, but I was introduced kind of piecemeal to the different aspects of it. So, like, oh, Mike, you might want to consider using an unbiased counting frame. Or, Mike, you might want to, consider using quantifying or selecting your fields randomly from from your region of interest. So sort of like these little piecemeal components that really are just the kind of logical elaborations of standard analytics that stereology evolves out of just naturally if you want to do, depending on what you want to achieve with the method of quantification or what your endpoints are, your ultimate goal is. But it didn't matter. So, with these effects that I was looking at, it was like an on-off switch in the brain.
So basically overdid it with stereology.
Yeah. So, and the whole goal with stereology is to kind of balance your sampling error against your biological error. And you want or not your biological error, but your biological variability. And you want your sampling error to be small enough that you can be confident whatever effect that you're seeing is due to the biological variability. And the impact that I was looking at, I could have introduced sampling error by throwing a dart, and I still would have found a significant effect, an accurate significant effect, because it was so profound. But then I went from there to Alzheimer's disease research. I was looking at transgenic mouse models of Alzheimer's disease, and I was doing stress. I was stressing them out. I was using a chronic variable stress paradigm, looking at the impact of stress on the pathology of AD in these mice, and then the treatment of that effect, trying to ameliorate that effect, with different compounds. So these are very subtle effects that you need an unbiased approach to accurately assess. And I was really focused on doing the behavioral work, the upfront behavioral work, the chronic variable stress. And that, for anyone who's done anything like that, is a very time-consuming, resource-heavy project. And so then at the end, you get all of your tissue. And then I started focusing on the analytics. But by then it was already too late. So I went in and I sacrificed my animals, and I got the brains, and I cut the brains, and then I mounted the tissue, and I did the staining, and then I sat down and I started figuring out how I was going to analyze them. And that it was, anyone who does stereology knows that it begins at the bench. It doesn't begin at the computer or at the microscope. And so really, I was kind of introduced to stereology more in that situation by, we had the scope, we had a representative from the company that we got our scopes from, and she came by about once a month to see how we were doing and how we were to help us troubleshoot the image analysis platform that we were using that came with the scopes. And she was very bright, and she said, this might be a situation where you probably wanted to use stereology. Now, she wasn't terribly familiar with it herself, but she was working with another lab that was. And so she would come back to me every month with a little more information from this other lab. And we started trying to do sort of a stereological analysis after the fact, which didn't go well. And so ultimately I had a very unproductive postdoctoral experience. And then from there, I went into contract research, spent a few years working.
That's where we work together now.
That's right. So before Charles River, I did some cardiovascular metabolic research, completely stepped away from neuroscience, was doing in-life work with rhesus macaques. And then I came back to neuroscience when I started working for Charles River, and I started working with Danielle Brown in Durham, who had been doing stereology for years. And it was my familiarity with stereology due to the failures that I had as a postdoc that really got me the job with her on her image analysis team. And then she sort of filled out the whole picture for me and put all the pieces together. And at that point, I was just completely hooked. And I'm a huge proponent of it.
Okay, so you basically overdid your first study with stereology. The second one you basically burned because you didn't do stereology.
That's right.
So let me ask you my first question. What is stereology? Can you tell us what stereology is in simple words for people who do some image analysis, or even a lot like me, but have never done this?
Sure.
What is it?
So I find that the best way to kind of explain it to people, it's really the logical elaboration of any quantitative analysis if what you want to achieve is statistically valid estimates of whole structures and not just a single sample from that structure. So as a scientist, I have some training in statistics, and
Me too, but I don't count on my training.
Well, one of the fundamental assumptions of any statistical test, and this is day one of Stats 101, is that the analysis or the sampling for the analysis was done randomly. And so when you you get a p-value, and from a statistical test, that p-value reflects that assumption that the sampling was done randomly. That's not always the case. In fact, sometimes it rarely is the case. It's sort of built into how people trim and section and sample from the tissues that they're doing their analyses on. You're really not using standard analytics, doing simple two-dimensional morphometry, not generating a p-value that is reflective of differences in the tissue that you sampled from. You're generating a p-value that's reflective of the piece of that tissue that you sampled. And efforts are in fact made in sampling from the tissue to equate samples across subjects as much as possible. So you try to get homologous sections, for example, if you're sampling from the brain. Because if you sample too far forward or too far back, then you're not comparing the same thing, because we know that the structures in the brain vary considerably across the rostrocaudal axis. And so and the same is true if you're sampling from even a more homogeneous tissue that has maybe different cellular populations. Then, if you section from two different parts of that tissue, you want to make sure that you're sampling or that you're making measurements on a homologous population of cells within that piece of tissue. Otherwise, you're comparing apples and oranges. The thing about stereology is you want to make these statistically valid conclusions about the whole structure and not just that single section, okay? And too often people will take the results that they get from that single section. They may say, oh, well, here we have a p-value of 0.001, okay? And we captured it from, this level, this bregma level in the brain, with these anatomical landmarks. And so we're going to say, and these measurements were done in, the hippocampus, or we did some linear measurements of the corpus callosum, or, measured the cellular population or the cellular density of a nucleus in the hypothalamus. But you only measured it at one level. And the conclusions that you're drawing are about that whole nucleus, or about the entire corpus callosum, or about, all of the hippocampus. And if you've only done two-dimensional morphometry, then anything that you say about that whole structure based upon that analysis you did on homologous sections is only speculation. And that p-value that you generated is not relevant to the whole structure. And so
I think an extreme case is when you are estimating immune environment of a tumor, where you only have a core biopsy of this tumor that is not even able to have a homologous anything of a tumor, because every tumor is difficult. That's a super important point. So let me just take one step back and put it in a super simple language with visuals. So let's say our head is the brain. Homologous sections of your and mine would be if we take a section here above eyebrows here, and with you above eyebrows, and non-homologous that is being done, but where you refer the analysis and p-value would be a speculation. If I take my section here and you would take your section here, and we compare and we say that we're comparing two brains, but we're actually comparing two different areas and they're not comparable.
Well, that is true, and that's sort of the trap that you fall into when you do two-dimensional analyses. Is if your sections aren't homologous, no conclusions that you draw are valid. So even the conclusions that you draw aren't valid for the sections that you've sampled at, because you've sampled from two different areas of the brain. What I was talking about before actually would be if, I sampled at my brow level and you sampled at your brow level, so we had homologous sections. Any conclusions that we draw are only relevant to brow
this
level.
Region.
We can't make any, and too often people will speculate and draw conclusions about the results that they get at brow level to, more apical areas of the brain, for example. So, and that's just not, it's not statistically valid. Now, in a homologous brain structure or tissue, where there's not a lot of variety across, the expanse of that structure, those sorts of conclusions, may be more acceptable. You can get away with that kind of thing. Similar to, what I was doing as a grad student, where, there were definitely some sampling error there was some sampling error, but the effect was so profound that anyone who looked at the images was just like, okay, yeah, clearly, here the subthalamic nucleus is activated, and here it is not activated, and where he sampled from really isn't important. If there is some heterogeneic expression of the biomarker that you're looking at, for example, which we see more and more as people do more and more molecular phenotyping.
So that brings me to my second question. When is it necessary? You said your first study, the effect was so profound that it was not necessary, whereas your other study, your second study that you burned without stereology, it would have been beneficial or even necessary to see anything. When is it necessary? What is it used for?
So it's really necessary if the target that you're measuring is heterogeneous, if it's heterogeneously expressed across your structure of interest, or if you're doing some kind of more,
which is like always almost.
Yeah, well, and increasingly so, because people are doing more molecular phenotyping, and now we're getting into this era where people are hyperplexing their data, and so they're doing you've got various profiles for all of the cells within a piece of tissue that are heterogeneously expressed throughout that piece of tissue. And so you have to ask yourself, when you're thinking about the analysis that you want to do, if I took a section from the tissue that I'm analyzing and compared it to a section from the same tissue from a different animal but taken at a different level, is the fact that I took those two samples from different levels of that tissue going to confound my ability to make any conclusions about the presence or the
absence
quantification of my endpoints between those two animals? And if the answer is yes, then you should use stereology.
Okay. So let me tell you something, an experiment that I saw on a 2D, 2D image that kind of is a lower level of what you're saying about generalizing across the whole structure. So same thing I just mentioned, the core needle biopsy of a tumor compared to a section of a tumor. So a core needle biopsy would be like stripe the thickness of my finger, and the full one would be my palm. So when you put this finger and let's imagine this is how a needle is directed into a tumor. And we did this experiment at some point and just directed it all around the clock, 360 degrees. Then like none of these were representative. And this is what's happening in medicine. You're drawing conclusions with this little finger compared to a full palm, and we are talking about both fists together. So imagine this, I don't know to which power that would be, but that what kind of reminds me of that. Okay, if you want to really have a conclusion of the full structure, stereology is the way to go. So that brings me to my third question. Why do scientists decide against stereology even when they shouldn't? And it's, not some scientists. I have never done stereology, and I'm now coming up with an example. Hey, we should do this. Why are we not doing it?
Well,
What's the problem there?
It is more time consuming, and it will slow your project down. Now, ultimately, stereology is really a time-saving device to, having to do a exhaustive count of objects or an exhaustive sampling of a tissue to get, the number of objects within that tissue or the volume of that tissue, or to measure the surface area of objects within that tissue, or to capture the local volume of objects within that tissue. So these are all things that you can do with stereology. You're estimating three-dimensional endpoints using two-dimensional samples. But
quick question.
Yeah.
How many of those two-dimensional samples do you need? We just said one is not enough, and we're trying to do with one. How many do we need?
So that was going to be my next point. Is so you're really increasing the number of samples that you need by about an order of a magnitude. So you're going from one sample to about 10 samples per tissue. And in fact, as opposed to the two-dimensional morphometry where you're trying to equate your levels and your sampling so that you're measuring the same thing from the same place, you actually want to randomly sample through the structure if you're doing stereology. So you're definitely trying not to capture homologous sections when you do stereology.
Oh, okay.
The principle that you need to follow is the whole structure that you want to draw conclusions about has to be present, and all parts of it have to have an equal opportunity to be sampled from each animal. And so what you'll do is we use something called standard uniform random sampling, where when we capture our sections through a piece of tissue, where we'll do a random start, we'll determine how many intervals we need or what size of interval we need in order to get anywhere from 8 to 12 sections from that piece of tissue. And then we will using a random number generator, figure out, where that first random start within that first interval will be. And then from there, we do regular intervals. So then every part of that tissue has the potential to be sampled from, but each animal is being sampled differently. And this is, I think a great way.
Let me show on my head. So let's say my hair would be this first interval where you randomly generate, and then for each animal it would be a random thing, right?
Right. So if that
here within this, but then let's say we take 2 fingers.
Yeah.
And then we go to fingers, to fingers, to fingers, to fingers, to fingers, to fingers, to fingers. But in each animal, the 2 fingers start at different point of my hair. First animal would be like at the top, and one of the animals could be at the bottom, anywhere in between.
Yep, you got it.
Okay.
Yeah, you're talking.
See, I'm
used to thinking transverse sections or maybe sagittal, but very rarely have I ever done horizontal sampling. But yeah, I mean, more people should do horizontal sampling. But yeah.
So does it matter like what kind of sampling, or is it determined by whatever the shape of the organ?
Yeah, it does. It depends. So again, not only do you want to randomize the levels that you're sampling.
Oh, you would have to do like this as well.
Well, if the objects that you're measuring, and if you're doing morphometric analyses, if you're doing like a volume analysis or like a surface area analysis of objects within that structure that are not isometric uniform. Okay, so that is, they're not, if the objects are already randomly oriented within that section, then you can go ahead and do like transverse sections throughout.
Okay.
But if they have some kind of fixed orientation within the structure, then you have to randomize how you sample from that structure so that you have an equal probability of capturing those objects at any orientation.
Okay.
So it can get a
it's
little
not
more
only the distance, might be orientation. So like basically 3D randomness you have to
That's
introduce.
right. Yeah. So and that's what I mean.
Yeah, I can imagine why scientists want to avoid that if they don't have to.
And you have to really get involved with your trimmers, the folks in the histo lab. They have to be trained in these methods of sampling and know what they're doing. And if you miss that opportunity to do that and the tissue gets used up, then you can't do stereology anymore, which is effectively what I was trying to do as a postdoc and wasn't able to achieve. But
in the second study where you tried to introduce this after the trimming and everything was done,
it was too late. Yeah. So it really you want everything to be random. You want to be everything to be random and unbiased.
Okay.
And so those are the assumptions that go into stereology. And if you can achieve that, and you've done that, then you can use a numerical metric to assess the amount of sampling error versus the amount of biological variability. And so and we call that metric of sampling error the coefficient of error. And depending on what analysis you're doing, what endpoints you're measuring, the calculus for the coefficient of error is different. And that's the hard math that goes into stereology. The estimate itself is actually pretty easy. You're really just keeping track of your sampling fractions all along the way, and then you're doing a supervised manual analysis, and then you're taking whatever values you get from that supervised manual analysis and extrapolating them based upon the fraction of the sample that you measure.
How does it make you feel when you see a project that is being done on 2D and that should have been done as a stereological project? How does it make you feel? And what do you think about the science that comes out of the project?
It's a little frustrating. I do have extreme, reservations about projects that, are not done using stereology when they should be. There are several documented examples of studies where not only can you lose the ability to detect a real difference, but you can actually detect a difference that's not there. There was a study of trabecular connectivity in mini pigs where using 2D morphometry, they found, I believe it was either an increase or a decrease in connectivity. And then when they did stereology, it was the opposite. It was not. So they found. Do you have
the reference? I can put it in the show video notes, in the show notes.
Several references.
Yeah, please do. Whatever references you have, we're gonna put them so that everybody can look at them. But like you say, I guess it's like with any scientific method. If this is not a method fit for purpose, then your results are like random. They're not reliable.
And I see it all the time. I mean, I think in the area that we're in, and I'm not a pathologist. But I see a lot of these standard analyses that are done in pathology that would really benefit from stereology, particularly.
I'm going to tell you something.
Developmental. Did you have something?
No. Go ahead, and then I'm going to comment on something that happened to me.
So particularly the developmental neurotoxicity studies and the ovarian follicle counts that are done are both prime examples of something that of analyses that should be done using stereology. In one case, you're doing object counting, so you would want to do something like a physical dissector and then use a proportionator or a meander random sampling for your area sampling fraction.
And we're not gonna go into the detail what these things are.
No, I won't.
Thanks for mentioning.
Yeah. So I'm just giving you kind of a primer, but that's some of the vocabulary. I won't go into the details, but and then for the other one,
I'm saying that because with this video, I want to give an overview what it is and when actually you need to go and dig out what are all those different methods that Mike is mentioning, so that when you get presented with a project and you see, oh, that should be stereology. Let me look into it before I start sectioning.
Here's a point that I want to make that I think it's a broad enough point that I hope people can appreciate, and that is this: because one of the things that has really come on strong lately, as you know, and I know, and I'm excited about it. I know you're excited about it, and our other colleagues are excited about it, is artificial intelligence.
Yes, this brings me to my fourth question.
Okay,
we both work in AI for. Let's call it flat image analysis, 2D tissue image analysis. But can artificial intelligence solve those problems, and how? I mean, I can totally understand if you don't have the money or the time or the resources or whatever, you just can't do stereology, then you basically count, okay, if the effect is strong enough, the results are going to be okay. So that's kind of your hope when you try to do a different method. But can we do better science with AI in this particular field?
In stereology?
Yes.
So yes. And one thing I do want to correct is that it's really not its own field. It's too often, I think kind of present it to people as a separate discipline. But it's, like I said early on, it's really just kind of the logical elaboration of what you're already doing if what you want to do is get statistically relevant, statistically valid conclusions of a whole structure. The eye-opening thing about stereology is, and I'm getting ahead of myself. Let me start with this, is what artificial intelligence is doing is it's introducing the ability to do consistent work at a high volume quickly. Okay. Mm-hmm. And that's really great. And shouldn't be devalued. However, the benefit of AI is getting conflated, I think, with the benefit of stereology, when they're really two separate things. And it comes down to the difference between precision and accuracy. So precision, AI can really help with that. That's, if you think of a target, that's the clustering of your data points, okay? The variability around the central tendency.
So it's like this flower here, that would be where they cluster or all in the center.
Right. So, well, like, and yeah, so like if the center, if you want to hit the center, you know, that's your ultimate goal. And that,
because that's precise and accurate.
That's precise and accurate.
Let's take this guy. Everything goes here.
Right. And so if everything goes up here on my shoulder and, you know, it's very precise, but it's very inaccurate. I've missed the center. And so you can have an algorithm that generates very precise measurements, but if you've introduced bias or too much error into your sampling, it doesn't matter because it's completely off target and you would never know it. And so this is the important thing with stereology. That's what stereology solves, is the ability to make accurate conclusions even when your measurement is imprecise. And that's been the really eye-opening thing about doing stereology, is just how imprecise you can be and still get accurate results. And so one of the, I guess, adages of stereology is do more, less well. And so there's this kind of trade-off where you can get very good results, but you don't have to be super precise. As long as your measure of central tendency is around, you know, the true mean, then you've got good results. And that's what the coefficient of error that I was mentioning earlier kind of measures, is how much work you actually have to do to get accurate enough results to be able to be confident in any differences that you find. And when you do that math, it's really surprising just how little work, I guess, you actually have to do. And you do this, you know, with a probe value, but I won't get into that. You're basically comparing your coefficient of variability to your coefficient of error, and you want your coefficient of error to be so many orders of magnitude, lower than your coefficient of variability. But
I assume you know these details, right? I'm asking because maybe we need to do a follow-up video on this one.
No, I do.
To go actually deeper into that stuff.
Be happy to. I think there are, you know, better resources for that than myself, but I love talking about stereology and learning more about it, because it is, you know, every kind of measurement that you want to do is has a different sort of approach, a different and there's a lot of different probes that you use in stereology to find what you want from your data set. Most commonly we're doing object counting and volumetry. But you can also, do measurements of surface area and measurements of length. And those are also very informative, particularly in tissues like, yes.
If you guys are watching this video till this moment, it means you're really interested in stereology. So drop a comment down below and let me know if you want Mike to talk a little bit more about this. And you see, he can talk a lot about this. I kind of have to tell you, you kind of disappointed me with your answer, because it still means we have to do all this work up front, and AI is not going to save us, or am I wrong?
this was what I was hoping to get to, and I hope
Yeah, you did.
I was rambling too much, but it is there. What we haven't seen is this merging of AI with stereology. And the opportunities are out there because people have digitized stereology. There are companies that have digitized stereology, and so now you can work and do stereology in a virtual environment. Now all you have to do is apply that AI to the stereology, and what you can do is really reduce your time cost. If you could, for example, do object counting. So basically the premise of object counting, if you've done everything correctly and you're randomly sampling and you're using an unbiased counting frame, is you're looking at, two adjacent sections, two serial sections. And one of the critical things in object counting is removing from your estimate double counts. Okay. And you're doing that in the XY plane, and you're doing that along the Z axis. And you've got different tools to be able to do that and different approaches to be able to do that. But to do it along the Z axis, you're comparing these two serial sections, and you're only counting objects that are present in one section and not the other. Okay. So then all you really have to do, if you don't want to do that manually, is generate an algorithm that can detect the presence or absence of the object and make that comparison. So then it's, and then only count it if it's present in one section and not the other. And that doesn't seem to me like it would be a huge leap from where we're already at. And then another example would be if you're doing volumetry. And then also the benefit of that is that then you don't even have to randomly sample from your section. You can count the entire region of interest, and you don't have to limit yourself to like 10% of the section, for example, which is what you commonly do in stereology. And that so you can go faster and do more work in less time to generate even more accurate results. And the same is true of volumetry, is if you can develop if you're trying to, get the volume of the corpus callosum, okay, through the brain, and you've got all your samples, and now, instead of using these point probes, which is what you would normally use, you do sample 100% of the corpus callosum, but you're using these point probes that have a associated area with each point probe, and you're really just counting points that fall on the corpus callosum, and then you're summing the area per point across everything that you've counted. Well, what you ultimately end up with is really the measurement you're taking is a very pixelated version of the corpus callosum. Well, if you have an algorithm that can just identify the corpus callosum and measure the area of the corpus callosum on that section, then you can have, a much higher resolution, where essentially each pixel is a point, and each the conversion, the area conversion for each pixel is your area per point. And then you just do your math, and you run it, and you don't have to do the point counting anymore. You can just
let me translate it and tell me if I understood it correctly. Let's take it from the beginning. We had the biopsy and the biopsy and the flat tumor.
Yep.
That was our flat image analysis. Then we take it to stereology, but we still are just having several of those flat sections, and we pick some points on those flat sections. What AI would give us would be, we have those several sections from our tumor, but we can do everything on this section.
That's right. And you could do it faster. And so why the heck not? And you're probably doing, when you do your probe analysis, which is what we do to determine if we're doing too much work or too little work, okay? If we're sampling, if we're being too imprecise, or if we're just being too precise, okay? Then, and normally if you get a probe value of above 4, you're doing too much. You don't need to do that much. And that's what I said earlier. That's the eye-opening thing. It's just how often you're just like, oh, I didn't realize I didn't have to do that much. And that's what AI is solving. I mean, everybody's putting their effort into AI, which is increasing precision. But what I've learned from stereology is you don't have to be that precise to get accurate results, as long as you're doing you're sampling accurately and you're doing or randomly and you're doing your measurements.
But this
in an
is
unbiased
something
manner.
this is a piece of puzzle that was missing for me always, because I was working a lot with annotations on flat samples, and I never knew, okay, how precise do you need to be with annotating this tumor? Is it okay if one pathologist goes like a millimeter below and a millimeter above? And I was looking for a way, some statistical way to tell me where is the interval of variability that the pathologists are allowed in this annotating. Does this relate in any way to stereology?
Yeah, it does. It depends on what you're measuring. So if you're doing object counting within that tumor, for example, you want to somehow, I mean, if you're doing object counting in that tumor sample without stereology, then that's really tough because you're randomly sampling and there's no real, and within a tumor, unlike with the brain, you can use anatomical landmarks in the brain to try to get homologous
Tumor,
sections.
there's nothing.
Saying, like if you're just doing a cut through like a peripheral nerve, for example, you can't get homologous levels from a peripheral nerve. It's very difficult and challenging to do that. So what you do when you sample from that peripheral nerve is you limit your analysis to, a certain population of fibers within that peripheral nerve. And you would do the same in a tumor where, we couldn't get homologous sections because really it's random anyway. They're randomly oriented anyway. So we're just going to limit our analysis to like a homologous population of objects within that tumor that have been, molecularly characterized. But even then, your sampling is biased. So you really do in that situation, or really should be using stereology to get your best results. And because where you're sampling from can definitely impact the conclusions that you're drawing and mislead you. So
thank you so much. This was a lot of eye-opening things for me as well, and I hope for you guys that this is valuable. Again, if you want to hear more about stereology, I'm happy to talk to Mike again, so drop me a comment below. And thanks so much for being my guest, Mike.
Oh, no problem, Alex. Thank you so much for having me. This was a pleasure.
If you're still with me, I think you're awesome. And it means you really are interested in digital pathology. So go ahead, subscribe, click the bell below to be notified every time I release new videos. Thank you so much, I very much appreciate it, and I talk to you in the next episode.